Hi, everybody. Hopefully you can hear us. This is Jeff Jensen. I'm here with Christian, and we're going to go ahead and get started. And if anybody happens to come a couple of minutes late, we will be providing a recording of this session so that people can view any parts that they may have missed. So this is a really exciting opportunity for us and a really exciting launch. The launch of the Ngs Kit is really a big breakthrough for us, and I think it's going to really help fire up IsoFlux sales and give all of you a great new capability to go out and promote to your customers. Um, it is really amazing that it's been a year since we launched the product. IsoFlux began shipping just about a year ago, and we now have 40 systems out there. And the expectations that we had when we launched the system have really been met, and those were that we were going to come out with a circulating tumor cell platform that did a much better job of recovering circulating tumor cells from clinical samples, and not just that, but also recover them alive so that they could be cultured, or that we could do RNA analysis, and that they would also be at high purity so that the variety of downstream molecular analysis techniques could be used as well. And finally, some other capabilities with respect to unlimited downstream options and the ability to use different targeted antibody strategies or cocktails to improve the ability to pull out circulating tumor cells, or give users the ability to select out a specific subset of cells that had a unique marker characteristic. And that's really been exactly what we intended from the start, and it has in fact proved to be the reason that people are adopting the platform. One of the issues, I won't really call it an issue, I mean, this was the expectation at the beginning, was that we were making an open platform that would allow just about any application, downstream application, to be performed. That could include counting, which was certainly the main application when we launched the platform, or the main thing that was being used on the most installed system, which was the cell search platform, but also other molecular applications like FISH analysis, single cell analysis, mutation detection, and other applications that were perhaps a little bit different as well. So those were all good things as well, but what we found as we're trying to grow the business now and grow the sales into new customers is we really don't have a killer app, and we want to do something a little bit different that allows us to go after and really focus on a very specific application that can really be our mainstream application. We also have had some issues with some of the ways that customers were validating the platform. We're getting caught in what I will describe as some painful CTC counting battles with competitors. And this is a challenge for us because we intentionally didn't design the system with counting as the primary endpoint, and we didn't develop a counting capability in the platform, which means that we are relying on the capabilities of the staff in the customer labs and the equipment that they have, and the capabilities they have are really all over the place, and in some cases they just don't have the necessary tools in their labs to do that effectively. Also, because of the nature of the system that we're selling, and the fact that we don't have an end-to-end complete workflow, it has tended to limit most of our sales toward what I'll term CTC gurus, people that are already focused on CTCs, and they have unique demands for their applications, and often those applications are unique to what they're doing and aren't necessarily broadly applicable. So we wanted to change that. We wanted to do something that would allow us to focus on applications that could be more mainstream and that we felt were really high value. So what we did is, about 6 months ago, we sat down here, and that was Christian, the CTO, who's sitting here next to me. Say hi, Christian.
Hi, guys.
As well as Mike and the rest of the technical staff, took a step back and said, okay, let's go back to first principles here and really look at what we're trying to achieve. And we took a step back and started exploring in more detail what the real goals were with respect to cancer. So first thing we did is we looked at what are the major applications in cancer. And I think it's important for us to all to go back and remember that the number of new cancer patients is quite significant. And this is a breakdown of cancer by type in the U.S. In 2010, the incidence by type, and you can see that if you sum all of these are the major categories. They add up to about 1 million new cases in 2010. Obviously, that's just we represent about 5% or less of the global population. So the number of new cancer patients globally is pretty astonishing. And if you think about it, Christian pointed out that within the U.S. We have a population of 300 million people. That means every year, 1 in 300 people is being diagnosed is a new cancer diagnosis. So that's quite remarkable that the prevalence is that high. So first conclusion here is cancer is a pretty significant cause of both disease as well as you can see, as well as death. However, when you look at the cancer drugs, 75% of cancer drugs, that's the bottom category, are not going to be effective for the full population. So only 25% of the patients are going to be responsive to a particular cancer drug. So there's a huge population of people getting cancer, but the drugs are not effective for the majority of that population. Third point, cancer is a genomic disease. And this is a quote from the physician-in-chief at Memorial Hospital in New York City, where he's saying that we know that cancer is a disease of the genome, and we're going to use genomic information and integrate that molecular data into our treatment decisions. So good opportunity here if we can figure out how to use our system to get at this genomic data. And in fact, that is happening, and it's happening in oncology. This is a chart from the McKinsey & Company that looks at both the potential for personalized medicine and on the y-axis, and the expected time that this is going to happen on the x-axis. And what you'll see here is, and it's probably not surprising that these two things are linked, the biggest potential for personalized therapy is in oncology, and basically the time to realization is basically it's happening now. So this to us says this is a great opportunity because the acknowledgement that the personalized therapy opportunities exist in cancer and is immediate. And in fact, the expectation is that next generation sequencing is going to be the dominant molecular analysis technique in many of the cancer types, including lung, colorectal, breast, and prostate, over the next 3 to 4 years. So it's happening right now. And so that caused us to think about, why is NGS the best tool? Why is that the way that this is going to happen? And the reason it's going to happen by NGS is that if you look at the number of potential biomarkers in the human genome, it's a huge number. So we looked at the COSMIC, which is the COSMIC database. That's the catalog of somatic mutations in cancer. And it lists more than, and this database is growing all the time, it lists more than 50,000 mutations that have been associated with cancer. So that's a huge number. Now, if you look at the commercial panels that are available, and we've worked closely with a couple of the AmpliSeq panels. Those are available from formerly Life Technologies, now Thermo Fisher. Those have numbers as high as 2,800 to 18,000 cosmic mutations. So they are available panels that cover that many mutations. Other techniques, other analytical techniques like qPCR aren't nearly as broad. You're able to look at typically less than 10 mutations, and if you really go to highly multiplexed PCR mutation panels, they get up to a max of about 100. So, given the number of mutations that are associated with cancer, it really says the only way that you're going to be able to do that effectively is using next generation sequencing. The other benefit of next generation sequencing is that it's benefited from an amazing development race that's been going on. And the dotted line in this graph is the predicted reduction in cost by Moore's law, which applies to semiconductor processors, microprocessors. However, you can look at the same kind of thing for any analysis, which basically is that you're going to reduce the cost and increase the capability on a order of, I guess, what is it, about every 12 to 18 months, you're going to cut the cost in half. Yeah. If you look at the cost to sequence the genome, it's actually followed Moore's law until 2008, and then it went through a dramatic reduction in cost by virtue of some of these newer next generation sequencing platforms being introduced from Ion Torrent and Illumina that have really driven the cost down just amazingly in the last 5 years. I do want to point out that this graph is for whole genome sequencing, and so the current cost for whole genome sequencing is somewhere in the $5,000 to $10,000 range. However, when we do a targeted cancer panel, the cost is much lower than that, so it's in the hundreds of dollars typically. So what's really impressive or really important for us to remember is that cancer is also a dynamic disease. So you're probably familiar or aware that many cancer centers are now using next generation sequencing to sequence a primary tumor biopsy. But we need to remember that cancer is dynamic. It isn't static. It is always evolving, and it's evolving in response to the environment. It's evolving in response to the way that the patient is being treated, and it's changing. So if you only look at a snapshot of kind of the primary tumor at the beginning of treatment, that's giving you a picture of the way it looks at that point, but it isn't telling you how the cancer is evolving. So it's been recognized, and as you can see here by Howard Scher, somebody that we've collaborated with, was recognized even back in 2003 that cancer is dynamic and it changes, and we need to monitor how it changes over time. However, the challenge is that if you look at the current diagnostic paradigm, we do NGS on the primary tumor at the beginning of diagnosis. Then the patient is treated, and that treatment might be surgery, it might be chemotherapy, it could be radiation, it could be a monoclonal antibody treatment, or some other targeted therapy. And the patient hopefully is going to respond to that. But in many cases, and probably about half the cases or more, the patient develops a recurrence, the patient develops a drug resistance, or the patient develops a new mutation. And the problem at that point, as pointed out in this quote from Thomas Cron, one of our customers at Bayer, is tissue is the issue. They don't have access to tissue to be able to diagnose what has changed in the cancer to be able to give the patient the right treatment at that point. So that's where we came to the conclusion that the biggest unmet need in cancer is patient monitoring. And it's a big diagnostic challenge because of the lack of tissue access and the fact that the cancer is changing, and that there's a potential need to look at thousands of possible markers that may be active. And we looked at that and we said, okay, this really is the opportunity for us. NGS has really come to the forefront. It's a mature analytical technique, and it's being used extensively for analyzing markers and determining the appropriate treatment when the patient has a solid tumor for which they can do a biopsy. But down the road, after that tumor typically has been removed by surgery, or it's been treated by a number of different therapies, there's no longer access to the primary tumor. The clinician knows that the cancer has spread, There may be micrometastases, small sites of microtumors that are spreading in the body, but the clinician does not know which markers are active, which mutations are active. The clinician only knows whatever I've been using to treat the patient is no longer working. So we looked at that and we said, this is the opportunity for IsoFlux is in patient monitoring, but there's a technical hurdle, and that is that to do next generation sequencing, we needed to get to higher purities of cancer cells from blood than we were able to achieve. As most of you know, we're about a 1% purity typically from the current IsoFlux approach, and we needed to get above 5%, preferably above 10%, to be able to make NGS work. So the good thing is that we embarked on doing this. We started a technical process to develop a way to get to higher purity levels than we could from the standard NGS kits, and we've pulled it off. And that is the IsoFlux NGS kit that we recently launched and that we're talking about today. The kit includes purity enhancement columns, so they take the output from a standard IsoFlux run and further enrich the sample. We've developed a new technology called bead activated size exclusion, and essentially what we do is we take advantage of the beads that we use, the magnetic beads that are bound to the cancer cells. They are 5-micron beads. So when we add those beads to the cell, the effective diameter of that cell increases by quite a bit and makes the cell plus beads quite significantly larger than any white blood cell is. So we use this bead activated size exclusion to retain the cancer cells that are bound to the beads and pass through the white blood cells. We then lyse and recover the cancer cells, and then go through a WGA amplification using the QIAGEN Repligy amplification kits. The resulting sample from that is ready for NGS library preparation and running as a standard NGS sample on the major next generation sequencing platforms. Christian is going to talk a little bit about the work that we've done specifically with Ion Torrent in validating the complete workflow on the Ion Torrent PGM. However, the same capability will also work on the Illumina platforms as well.
Thanks, Jeff. Hello, everybody. So first of all, I wanted to point out that you, as questions come to you about the presentation, you should not forget to write them either in the questions section or there's a little chat section on your GoToMeeting console. So please do that. And we had another presentation here. We got a lot of good questions, so I'm hoping the same will happen here. So first off, I wanted to talk a little bit about the purities that we're obtaining now with this new NGS kit. And this is the analytical portion of that work. And so what we're doing here is we're spiking in MDA and B231 cells. This is a mesenchymal line breast derived cell line, breast cancer derived cell line. And yeah, and what's happening here is we are going through a full experiment from the spiking, as little as 10 cells are put into a blood tube, all the way up to 200 cells, through the IsoFlux isolation, and then through the next-gen sequencing high purity enrichment column. And here you see that we're getting much better purities than we would normally get. Most of them are greater than 5% for 90% of the samples, and a lot of you have probably done these experiments before, and so you know that normally we were more closer to the 1% range. Okay, now the most important thing here is to have this process be workable for clinical samples. And so what you see in this next slide is our clinical validation data set, where again, we took clinical samples from a number of different indications. We put them through the process, and then we counted both the background and the target circulating tumor cells based on CK positive, CD45 negative cells. And again, of the samples that were CTC positive, we found that 80% were above this 5% purity mark. So again, we were really happy with this set of results. Now, a lot of our work, aside from developing the platform, has been really done in conjunction with IonTorrent, who's geographically very close to our offices, and they're one of two leading next-gen sequencing platforms out there. And in conjunction with their team, we've developed this workflow that starts really with the blood tube sample and goes all the way through to determining the variants present in a tumor sample, in a blood sample that is the variants that are tumor derived. So again, the process starts with a blood tube. We typically use 7 to 15 mil. We recommend a larger volume that can go into a single, up to 15 mils can go into a single IsoFlux cartridge, and we recommend using 15 mils, but we've validated this down to a single tube. We then perform the immunomagnetic enrichment, followed by the next-gen sequencing kit, which is, again, a benchtop process that follows the run. And then as the analysis was done on the PGM using the AmpliSeq hotspot panel. So I should point out that a number of different panels are available and can be used successfully. We've actually done validation for 3 different panels using this process. One is a custom panel from a CRO called Assuragen that has a smaller number of genes. The AmpliSeq panel is the standard IonTorrent cancer panel, and then there's also they offer what's called a comprehensive cancer panel, which is a larger panel that in this case has about 400 genes compared to the 50 genes of the AmpliSeq. A lot of the data I'll show you here was obtained on the AmpliSeq. So we focused on that workflow because that's the most widely used set of somatic detection amplicons that is run on the IonTorrent. And so again, recapitulating the workflow, we have a part of the workflow that's done on the IsoFlux that goes from the starting sample through enrichment, and then what the Ngs Kit does, besides increasing the purity level and decreasing the background, it also performs WGA, so whole genome amplification of the resulting DNA, and it ends up with a sample that's ready to be put into the sequencer. That goes into the IonTorrent. Because we're using a AmpliSeq library, we have to go through the library prep first, then the NGS, and then the variant calling and analysis. And for the variant calling, what we've developed is a set of variant filters that will result in high confidence variant calls that are very sensitive down to a 1% allelic frequency. And so basically what's shown here is a screenshot from the Ion Reporter cloud-based software package that's available to all IonTorrent users, and it shows the resulting 3 variants here. There's a BRAF, a KRAS, and a TP53 variant from one of our samples. And then to the right, you can see the actual filter chain. And we don't expect you to take all of this in, but just bear in mind that if your customers want to do this, we have a very well prescribed and well-validated method that gets down from the total number of variants, which in this case was 31, down to the ones that are high quality and they're not germline but somatic variants. So in this case, there's 3 of those. Okay, now also very important, a piece of the work that we did here was we sent the same samples out to 2 different sites in order to basically validate that independent sites could perform this workflow and come out with the same result. So for this experiment, again, we used blinded samples and we went start to finish, and we started with these MDA cell line that was spiked into blood, and then the samples were sent to one site was Iontorin, the other site was a core lab that's nearby. So basically, what you see here is that the cells have these three variants, a BRAF, a KRAS, and a TP53 variant. These are classic mutation hotspots for a number of different cancers. And then you can see the purity level. Now we took one of the samples was enumerated, and so that yielded a purity level. The other two samples were put independently through the workflow and then split up and sent to the two different laboratories. So you can see that there's really, first of all, there's good sensitivity. So you see at the lowest spiking level, which was 2 cells per milliliter, you have a 5% resulting purity. For the heterozygous mutations, you have a 2% allelic frequency, and we see all of those variants in both sites. And then the other thing that's important is the false positive rate is very low. So you can see number of false positives that come through our filters are also plotted here. And really, we only got one false positive for one of the normal samples in the second site. So that is very good performance in terms of being able to determine these calls. And here you have the basically comparison of the variant calls for the two different sites. So you have 3 different genes here. Each one has a specific single nucleotide polymorphism, and the allelic ratios for those at site 1 and site 2 are plotted here. So what you should see is really nice concordance. The R square here is actually 0.97. So let me just give it back to Jeff here, but I think that what we've got here is a product that's extremely unique, and we're very excited about this development. We think NGS is the future of molecular analysis, and now with this new kit, I believe we're the only CDC platform that can take the whole output of the platform and go into an NGS reaction without doing things like single cell picking. And it's also really helpful that we're working directly with one of the major providers of NGS platforms.
Yeah, a nice thing about this, obviously, also is from a sales standpoint, your addressable market can be anybody that's doing NGS and anybody that's doing NGS for cancer, and obviously there's a huge and growing installed base of sequencers out there that are being used exactly for that. So I think it's a great built-in opportunity to add IsoFlux to many of the systems that are out there. We'll also be doing co-marketing with Iontorin, so we're going to be putting out some technical content and webinars. We'll be presenting jointly at upcoming genomic and cancer conferences, and they'll be communicating this out to their customers as well. So I think that's going to provide us with some good lead generation. And of course, the fact that we're doing this in a partnership with Iontorin, part of Thermo Fisher, is great from a technical credibility standpoint as well. So just so you have a handle on sort of the kind of costs that we're looking at for the entire workflow, this doesn't include labor, but it does include all the kit costs. So IsoFlux, you're looking at about $300 for a combination of the IsoFlux standard recovery CTC kit, as well as the NGS kit. This would be kind of at list prices to the customer. So obviously, your distributor cost from us is going to be lower than that. And then the AmpliSeq Hotspot Panel V2 is probably in the $300 to $400 range for the chip and the reagent costs. So you're talking about a total kit reagents going from a blood sample all the way to getting next-gen sequencing data of roughly $700. So it's a pretty cost-effective, given that you're looking at a total of 2,800 mutations. One of the nice things about this is that this can really be a universal assay. So yes, certainly users can come up with targeted panels that might focus on a particular indication like breast or lung cancer, but it's becoming so cost effective that looking at a kind of a pan cancer panel becomes affordable. Second important thing here is we have historically been selling IsoFlux just to people that have been pretty much self-identified as interested in CTCs, and they can be great customers, but they can also be very challenging because their applications are very demanding. So one of the things that we're really excited about with the NGS capability now with IsoFlux is it opens up a much broader range of customer prospects to us. We don't need to be looking at people that have indicated that they want CTCs. What we can look at now are more broadly any customer segment that has an interest in monitoring patients for cancer markers. And that can be pharma and biotech companies that are developing drugs for cancer and want to be able to monitor patients in a clinical trial, or ultimately be able to develop a companion diagnostic for their drug. And where we're seeing, and certainly we're seeing adoption there. We're also seeing a pretty significant adoption at cancer research centers. So we're working with groups like MD Anderson and Stanford and UCSF and Moffitt Cancer Center here in the U.S. that are really excited about. They're already doing sequencing of tumors for cancer patients, and they recognize that they have a real gap in their capabilities for patients that are showing that they're becoming non-responsive to treatment or are recurring or are going metastatic, and they don't have an ability to run NGS on those patients, and they're really desperate for that. So it's a great new capability that they're really excited about adding into their menu. And then finally, we're also working with molecular diagnostics labs and CROs. Here in the U.S., we have a regulatory path for lab-developed tests, and we are having these molecular diagnostics labs and CROs adopting IsoFlux to take it through that path. Other countries, in some cases, have access to similar regulatory pathways where the lab can develop and get its own test approved. And we certainly want to look with you at each of those specific cases to see what's appropriate in your particular geography. Regardless, one of the important things here is if you look globally, there were probably hundreds, maybe 1,000 CTC researchers. When you start broadening the addressable market to include anybody that is interested in researching or developing a diagnostic that's able to look at cancer generally, the addressable market becomes much, much larger. So we're excited about the fact that we're going to be able to broaden our scope pretty significantly to the broader cancer community at this point. So how are we going to do this? Obviously, we're going to start putting out a bunch of promotional materials that we're going to make available to you. When we did the session this morning, I already got requests immediately for collateral that could be used in mailings that the distributors plan to do as soon as possible. So yes, we will be coming out with collateral that you can use to do your own mailings. As you know, we already have a data sheet that's out. The next thing that we're putting out is an application note with Ion Torrent, and that, in fact, Christian is over my shoulder editing it right now. It looks really impressive, and I think that's going to be a great both technical piece as well as a promotional piece that you can use. And then initially, that data sheet or that app note rather is only going to have analytical validation data in it, and we want to put it out with that. We don't want to wait. But we also are, of course, working with clinical partners. We're working with a clinician at UCSF and others, and we're going to be adding to the data sheet or the app note rather clinical data as well. So it'll come out first with analytical validation data, and then we're going to follow that up with clinical data as well. We'll also have a webinar along with a customer, and that'll take place in September. And then there are a number of conferences that we're going to be attending and that we, along with Ion Torrent, have submitted abstracts to. So not only will we be in attendance at these conferences, but in most cases we'll be presenting either a podium presentation or, at the very least, a poster. Along with this, we also took a step back to take a look at how we were doing demos. And I'll be honest with you, we've had some challenges in the field with doing a complete staining and counting workflow for clinical samples at customer sites. And our conclusion is it hasn't worked very well, and it's not something that we want to continue. We found there's just too much variability in the capabilities of the staff at these labs and also in the equipment that they have available. We consciously made a decision not to offer complete counting, and that was by design. We meant to do that because we don't think it's clinically meaningful, and in fact, more and more data is coming out bearing that out. But if we go in and do a head-to-head counting demo against cell search or any other approach, it just becomes really painful. It's not an analytical direction we want to go in, and it's hard to do in the field. So it's really not something that we want to continue to promote. So what we need to do is we need approaches that demonstrate our superior performance of IsoFlux, that demonstrate our NGS capability, and don't focus too much on counting. So this is what we propose. We've had really good luck with counting pre-stained cells. And I know that this is something that a lot of you have used and have had good success with. It works well. We recommend that this be done with pre-labeled MDA MB231 cells. This is the mesenchymal-like cell line that Christian talked about earlier. It's known as a challenge to isolate because it does not have very high EpCAM expression. So it's a great validator of our isolation and recovery capabilities. It also has known mutations, so we can actually take this one all the way through the NGS process as well. So number one, continue doing that. We think that's a great approach, and it demonstrates the basic capabilities of IsoFlux. Second thing we can do is we can also provide amplified DNA to you as a reagent effectively, and this is basically it's basically spiking in the MDA MB231 cells here. We run that through the full process. We run that through the NGS kit so that we end up with amplified DNA ready for sequencing. And what we can do is we can offer that as a reagent. It'll have a nominal cost associated with it, and you can buy that from us as a reagent that you can give or sell to your customers to have them try on their sequencers to validate that the full process works. We can also have you process samples locally. So if you have customers that run samples on your demo IsoFlux system, we can have you send the sample back to us, and we can run it either for counting or all the way through next-gen sequencing for a nominal cost. So that gives you an ability to run IsoFlux locally, send the samples back to us, and we then process them. The reason we want to do that is that we have better quality control here. We run these assays every day in our service lab. We do them for major customers like Bayer and Genentech all the time, and we have it reduced to a very well-honed process versus doing it in the field where it's a little bit subject to the vagaries of the skill set and equipment that the customer has. However, if your customers have ion torrent PGMs, you can run the entire workflow in the field. You can process samples on IsoFlux, run them through the NGS kit, and then have your customer run the next-gen sequencing. Currently, we'd like to limit that to the ion torrent PGM because that's the one that we really have the analytical workflow figured out on. We'll be adding other platforms to this, like the ion torrent proton and the Illumina MiSeq and HiSeq. But for right now, we'd like to limit it to the PGM because that's the one that we know works. So one of the nice things about this is it is a unique positioning. Nobody else right now has this capability. Nobody else has a complete workflow all the way from blood sample to next generation sequencing. So We think that selling against the competition is going to get a lot easier with this approach because we really have carved out a unique position. So utilize the new data that we're putting out, as well as the new demo workflow to show the superior capabilities of our system, both in purity of the sample that we produce and the ability to go all the way through a robust next generation sequencing workflow. That's something that only we can do, and I think that's going to be a major game changer when you go out and sell this. Finally, we're going to continue to develop new publications and data and utilize our partnership with Ion Torrent to continue to add more technical content that you're going to be able to show. And I think the fact that we've done this in partnership with Ion Torrent is going to be able to lend a lot of credibility to the capability. So we hear a lot, and it wouldn't surprise me at all if you're hearing this as well. We hear a lot about cell-free DNA as a competitor to circulating tumor cells, generally, and IsoFlux specifically. So why is it that we are going to be able to win and beat cell-free DNA? Well, this slide shows why. Because of the very low purities that, whether it's the cells, the circulating cells, or whether it's just cell-free DNA, the low levels of concentration that those exist within blood, unless you do a very, very, very good job at enrichment, you're really not going to be able to do very much with respect to being able to identify these low-level mutations that we're looking for. So with cell-free DNA, they're also forced to go to panels that only have a handful of markers. In one case, 47. In the case of Boreal, they claim 100, but we generally see them running 10 or fewer markers. So the challenge, the limitation right now with any of these techniques is that without dramatic enrichment, you're really going to be limited in sensitivity and only able to look at a few markers. So your major selling point against cell-free DNA is the fact that they're able to look at only a very small number of markers. And as we talked about, cancer is a mutational disease. It can head in many different directions. So you really don't want to limit the number of markers that you're able to look at beforehand. You'd like to look as broadly as you can. The way we describe it is you want to cast a broad net because you're not really sure what you're fishing for at this point. And the reason for that is that if you look at the relative purity levels of different samples, buffy coat, that is just the white blood cell fraction. The fraction of the buffy coat that is circulating tumor cells is 0.0001%. Obviously, that's far too low. Filters and cfDNA get you to about 0.1% purity, and cell search gets you also to about 0.1%. We're about 2 logs better than that. We're getting to 10% purity. That is why we are uniquely able to run next generation sequencing and the other technologies cannot. So cell-free DNA, the reason that they can only run a very small panel is that they have to do some sort of enrichment looking specifically for certain fragments of DNA that are already known that they want to look at. So they have to know what they're looking for going in. We don't. We can run a very broad universal panel that can look across all cancer mutations. That is the unique benefit of IsoFlux, and that's really what we need to go out and sell. So next steps, we're getting to the end here, so anybody that wants to ask questions, you can start typing now. We will confirm kit pricing for the NGS kit in the next week. We apologize for that. We're still finalizing some of the component costs. We will commence shipments of the kits July 15th. July 30th, we will produce the NGS application note co-written with Ion Torrent. We will be doing global email blasts with that material to our list. That is a global list, so it'll include some of your customers. And then in September, we will have our webinar with Ion Torrent UCSF. So what we hope is you can start with your own promotion and sales activities with the materials that we're going to put out this month. And then we're going to be bolstering that with other promotional activities beginning in August and continuing through the fall. So, in conclusion, we are really excited about this game-changing capability that we're coming out with. We've now gone from a sample prep capability to an end-to-end analytical capability that takes us all the way from a blood sample all the way through to next generation sequencing data from that blood sample. And we're calling that a liquid or a blood biopsy. It's the first true blood biopsy or liquid biopsy that has been able to be obtained from blood. And there are a number of applications that this opens up. It allows us to go after a much broader customer base, and it really creates a capability that right now nobody else has. So we have first mover advantage, and we really want you guys to go out and push this hard because we think this capability, as we have seen in our sales activities in the US, this capability is something that people really get excited about. As soon as you say we can do NGS of full cancer panels from blood, you're going to get ears perking up and you're going to get excitement. So this is a game changer for all of us, and we're excited to be able to introduce this to you, and we think it's going to be a great addition to your product line and your sales activities, and we're looking forward to kicking off the second half of 2014 with the launch of this, and looking forward to making it a huge success with all of you. So with that, hopefully you are asking us any questions you have, and are you getting a few, Christian?
Not yet.
Not yet. Okay, so
People are typing.
so we can give you a few. We can start off with a few that we got this morning.
Sure.
Okay, here's first question we got was, can we do RNA-seq as well?
Yeah, so RNA-seq is obviously a capability. Because we're starting with intact cells, we can extract the RNA, we can look at the expression as well as abnormalities, mutational abnormalities from RNA. This is not something that is as polished as our DNA workflow, but it's absolutely doable, and I know there's a lot of interest in that. Another question that we got earlier was, do we really, there was a question about counts. Do we really not want to do counts? Do we really want to, what do we do? In other words, if the customer does ask to have their clinical samples, counted as part of a demo. And I guess maybe Jeff has some thoughts on this, but basically, a good thing to do is we can do, as Jeff explained, we can do the spikes and we can do the counts. I think where we've seen things go wrong is if we don't have pre-labeled cells and we're trying to train the user to do the whole process, starting with spiking, but then also staining and mounting and imaging, everybody has a different set of equipment, so that's where things go wrong a lot. So we've had much better success where we've done the staining and the counting here at Fluxion, and so if people really need to do that, we can offer that as a service. We don't advise that you do that in the field, without having a very well prepared set of equipment and protocol that they can follow. Okay, just bear with me. There's a bunch of questions that all came at once. There was a question that said from Dr. Kim about how many white blood cells are contaminated. I believe you're asking what the contamination, the background level is. It's in the level of a few hundred. So you can imagine, we're achieving down to 20 cells spiked, we're achieving 5% purity and above. So I would say it's between 200 and 500 cells typically is the background. Okay. There's another question that says, what is the purity threshold for the Ngs Kit analysis? From Dan. basically, what we found is that for NGS on the ion torrent, we can detect as little as a mutant allele frequency that's as little as 1.5%. Okay. Now, our threshold for tumor material is higher, it's at 5%, and that's because not all the cells, not all the strands of DNA have that mutation. So you might have a heterozygous mutation, in which case only one strand has the mutation. So it's 50% of that. So 50% of 5 would be 2.5. Or you might have a situation where you have a mixed population of tumor cells. So only some percent of the cells have the mutation. So we think that the percent allele can go down to 1 to 1.5%. Percent tumor material should be above 5% for good success rates. Okay. Okay, there was another question from Rui about how the purity column works. Basically, what we've done is we've taken an approach that's called, orthogonal separation. So basically, we're using two different characteristics of the sample to separate. And one characteristic is the magnetic force, the fact that the beads are bound to the cells and they can be moved in a magnetic field. And the other, in the purity step, we're using the actual apparent size of the cell bead complex. So basically, when the beads are bound to cells, they increase the apparent size of the cells, and we use a second size-based separation to remove even more of the white blood cells that are in the sample.
How many tubes? That's another question we got this
morning. Oh, how many tubes? Yeah, so the another question we got this morning is, do you really have to use 2 blood tubes or not? And what we found is that if you use a larger blood volume, you do get more cells. You get twice the number of CTCs in the sample, and that's good for this type of analysis. The purity is also a little bit better, but not dramatically better. It's fairly similar to just using 1 tube. So because we have a minimal number of cells that we need to capture, you get an edge by using a larger blood volume. But absolutely, we can successfully process and have the right purity for a single tube. It's just giving us a greater chance to capture circulating tumor cells. I don't know. You want to add anything to that?
No, just and you run, whether it's 1 or 2 tubes, you run it through one of our cartridges and one of our NGS kits. So it doesn't change you're not having to run 2 samples. You're just running 2 tubes as 1 sample.
Yeah. The other question we got from Dr. Kim was, what is the sequencing depth for amplicon sequencing? That's a good question. We have used per chip, we have run between 10 and 12 samples with good success. At one site, we ran 12 samples a chip, and the other one 10. That translates to about 2,000 reads per site. So typically, you'll have, let's say, 2,000 wild-type reads, and then 10% of that is would be about 200 mutant reads. So that's the read depth that we use.
We set a minimum threshold?
There's a minimum threshold for detection. If you look at the one slide, or you can't flip through the slides, but if you look at the filter set, we have a threshold of a minimum of 20 mutant reads for a variant to be called in our protocol. Okay, thanks, Dan. Bye. Let's see. How can I send the samples to you for counting and sequencing? That's another question from Dr. Kim. So I don't know. Maybe Jeff, you can address that. I mean, it's just
we've had the problem. We get this all the time. We do get samples from Bayer, for instance, from Germany. So they can be shipped frozen and not a problem.
On the Buffy code form.
Yeah, Buffy code. You can't do that with whole blood. So the preference would be, in your case, would be ideally run IsoFlux locally, and then send us the cell pellet frozen. And we have instructions for how to do that. We have a protocol for that. So we would get you the protocol for freezing, and then you could send that to us, and then we can locally do the counting and or the next gen sequencing on, for example, the Ampliseq cancer hotspot panel V2.
The other option that Jeff mentioned is to do a phycol separation and then freeze down the bucked goat, in which case we do the separation here. And we've done that quite a bit too, and we have protocols for that too that were validated. So please, if you're planning to do any of this, please contact us so we can give you the right protocols.
Okay, we have another good question here, which is if we have prospective customers that are skilled at using Illumina, the Illumina platforms, and probably using like a TrueSeq panel, do you advise that we still approach these customers to use the NGS kit? And the answer is absolutely. The kit is absolutely applicable to Illumina's NGS platforms as well as Ion Torrent. The only minor point is, as of we have to go through similar. One of the things we didn't go into too much detail here is we did spend a lot of time with Ion Torrent developing the modifications to their analysis, their software analysis platform to make it work well for our particular sample type. Even though we've improved the purity dramatically, we're still at the lower edge of where these platforms tend to operate. So we need to do some similar stuff for Illumina. We are engaged in doing that with some partners right now. So we don't have it as fully baked as fully worked out as it is for the Ion Torrent systems, and we're in the process of doing that. So I would say for right now, you could probably be talking to the people, and by the time we would get to actually running a sample on one of their platforms, we'll probably have it worked out.
Okay, so there's another couple of questions. One is there a minimum number of cells that need to be captured in order to get good results after further NGS kit enrichment and NGS analysis? So if you look at the presentation, we got really good purity for all the patients that were, we consider CTC positive, and those who were people that had 10 CTCs or greater. So I think if you've got a minimum of 10 cells, you're in good shape.
Now, if you go back and look at our older data and look at 10 cells, you're going to get more than 10 cells. If you go across all the different cancer types that we've looked at, you're going to get more than 10 cells, probably about, I think it's about 70% of the time. However, if you use 2 tubes, that number jumps to greater than 80%. So I think we're in the greater than 80% range with 2 tubes.
And then there's another question about ClearBridge and how we compare to that product.
Okay. ClearBridge. So, the challenge with a lot of the other systems that are out there is we're seeing very little analytical data from them and very little clinical data. So often the companies are making claims and don't have much data to back it up. ClearBridge has a couple of different platforms. They have an older version that is kind of a chip-based technology where the cells are captured in slots that they believe the CTCs get caught in, and the white blood cells pass through. We've seen limited data on that. We believe the purity level is very low. So the knock against that would be, one, how do you recover the cells out of the chip effectively without major dilution, and two, low purity levels. They have a newer technology that uses Dean flow fractionation, which we know less about in terms of its performance. It is again using size of the cells as a way to isolate them, and every size-based approach we've seen suffers from low purity. So we expect that it's going to have similar low purity. So the comparison for most of the size-based approaches is that they have very low purity levels.
And just to be more specific about that, what we found in the literature, and if you need to look at this, we can send you references for it. The filter-based approaches where they have decent retention have typically about 10 to 30,000 background cells that remain on the filter, depending on the volume of blood used and everything. So that's kind of a couple of orders of magnitude off of where you need to be. Kind of reached an hour. Should we?
Okay, and a question, can you stop after enrichment to retrieve the cells before lysis? We got that question this morning as well. Great minds think alike, obviously. So the answer is it can be done. It is not I would say it's not straightforward and probably not recommended, but.
And I see the follow-up to your question, Agnes. I see that you're asking about single cell analysis, doing this for single cell analysis. What we found is that for single cell analysis, you don't need that high purity to be able to pick cells. You're enriching anyway by picking that single cell, and if you plate the cells at the right density, the picking is possible. And we actually just have a nice app note coming out on doing single cell picking from our enriched samples, but not using the NGS kit that uses both one automated and a manual set of picking results. So I think that's readily doable. I would advise customers not to use the NGS kit for that.
Okay, it looks like we've gotten to the end of the questions. If we missed any, absolutely shoot either Christian or me an email, and we will answer to the full group. We think these questions are great, and want to get them out to everybody. So feel free to send anything that we didn't get to, and I want to thank you for your attention. And it seems like there's a lot of excitement out there about this, and we're really happy to hear that, and look forward to working with you and making this a big success. So thanks, and have a great day. Appreciate it.
Bye, guys.
Bye-bye.
Kevin.